the first, I think it was evening, just reminded me the inaugural seminar, the IAS seminar series on big data, and this one is in cooperation between the IAS seminar series on big data and the Division of Social Science. And it's a real pleasure to welcome James Evans from the University of Chicago. If you read the bio attached to the talk announcement, you'll see that he's involved in very interdisciplinary work in housing sociology, but working with people across the University of Chicago campus in computer science. Even it turns out he knows a high school classmate of mine who's at Oregon in high-performance computing and so forth. I first became aware of James Evans' work. Actually, one of my last duties at UCLA was the chair of search committee, where we hired a postdoc of his who had actually earned a PhD in physics, but had transitioned into the study of social networks using some of the network methods that he learned in physics. And so I learned about James's work then, and again, very excited to have an opportunity to bring here to HKUST and hopefully catalyze some conversations across the different schools and divisions here at HKUST and how we can all work with each other on questions in big data. So, James.
Great, thanks. It's working. Yeah, pleasure to be here. As Cameron mentioned, I'm at the University of Chicago, and then I run a little center in our Computation Institute called the Knowledge Lab. So I'm going to be talking a little bit about what I'll say how science thinks, how science as an institution thinks, and how one might think about thinking better based on using precise instrumentation of limitations, biases, heuristics through which scientists search through this space. So it should be, oh, and I should just say, just as a matter of policy, I'm happy to kind of run through and share my thoughts and kind of wait for questions, but I love being interrupted. So, and argument is the center of all this, and we'll kind of break open the veneer. So feel free to kind of dive in whenever. So I'm arguing, and I think in some ways there's no argument here. The science itself is a complex system. It contains diverse, differentiated components that have strong interactions with one another, leading to emergent, often unexpected collective behavior on many scales, right? Leading to objects like this detector at the Large Hadron Collider, which is the result of the work, that really irreducible work of experimentalists and theorists and engineers and many others. There was an interesting piece in Science about 5 years ago by Richard Foote talking about mathematics as a complex system. And here's the root system of the exceptional group EA, but there are a number of cases in which these intersecting components of science, and not just the bodies, but the theories and the elements and the units and the theories themselves kind of bubble up and create unexpected collective outcomes. And so I'm going to be talking today about analyzing the system and then gaining some insight and intuition from the system about how to tune it for the purpose not only of identifying better pathways for individual science, but how to think of steering science as a collective endeavor from the perspective of science policy. My models of scientists and humans are necessarily somewhat impoverished. So think of the individual ant, right, responding to pheromones and collective natural environments, and yet through these simple interactions create these kind of works of majestic structure. And so the roadmap for today is first I'm going to talk about publications, scientific publications as a context and really a substrate for scientific thinking, which reveal kind of heuristic and biased reasoning pathways. The second I'm going to talk about, I'm going to use this as a context then to talk about the balance of tradition versus innovation in science. I'm going to explore the efficiency of reasoning in science. And then I'm going to leverage these at scale for a particular case in science that I probably at that point won't have that much time to talk about. So we're doing a number of cancer screens to identify optimal cocktails. And so using our understanding of the scientific system then to kind of derive ex-ante experiments in the context of the biosciences and cancer research in particular. So I'm going to argue for really the central role of publishing, both print and digital, in the way that science as a system thinks. And I'm going to use these deep stacks of back files that scientists themselves can read, but also using computation, natural language processing, named entity recognition, measures of information, graph, and probabilistic theory. You can link these things to tell broader narratives and identify regularities that might be unnoticeable individually, right? So you can link together from these papers authors, institutions which have geographic distance from one another, entities, methods that may or may not be independent of one another over these spaces, chemicals, other theoretical mentions, and you can identify these as elements in this broader kind of hypergraph of science. So chapter 1, right, I'm going to kind of explore the possibility that published science really acts as a neural net on which the system of science essentially walks and thinks. So scientists think through this complex network of context and content which you can kind of extract from these publications. And I'm going to use as a kind of a basic model this intuition from Newell and Simon from 1971, this idea that scientists as problem solvers really walk through a network of possible wanderings as they identify possible outcomes. And I'm going to consider them walking along the network of biomedical publications. So the United States National Institutes of Health kind of sponsors the National Library of Medicine. And so for this first little mini analysis, we're going to be looking at about 20 million papers from 1865 to 2010, about 10 million authors, 10,000 chemicals, 5,000 diseases, and 2,500 methods. And this data set reveals transformations in the institutions of science, right? So obviously, there's the growth of each of these things. But if you look within each of these nodes, right, you have methods becoming much more central as science becomes institutionalized. You have diseases remaining relatively stable, but a huge influx of chemicals and authors growing through this period, right? So you've got things as common as X-ray tomography and immunohistochemistry down to speleotherapy and a variety of very rare but identified concepts. Within particular papers, you see this growth from about 2 to 4 authors over this period, which tracks actually the use of methods which identify those authors. So scientists are effectively one-trick ponies over this period. But what you see when you think about the distance by which people could walk through the space of elements, right? So if I could kind of reach another author in this space as a function of us both using the same method or us both studying the same disease, that everything, because of the hyperconnections which are so dense, everything is virtually at distance 2, right? So anything that's not connected, so authors connecting the chemicals are at distance 2 from one another. And any chemicals that are not connected already are at distance 2, again, if they go through. So thinking about just simple graphs in this context doesn't make much sense because everything is so densely connected. We really need to think about the richness of these paths and identifying the likelihood of new papers emerging over time. So what I'm going to do in this first part is kind of try to model the degree to which past papers predict new papers. So consider this paper here. NAP protects hippocampal neurons against multiple toxins. So there are lots of things in this paper. There are a string of authors. There are chemicals, this fematomolar acting peptide, NAP. You've got kynic acid. You have macromolecules like this microtubule-associated protein 2. And then you've got phenotypes or diseases, in this case, cognitive function. And if you link all these things together, you can see essentially these papers as hyperedges in an evolving hypergraph, right? So a hyperedge is a set, basically, of these containing elements, right, of authors and chemicals and methods, and in this case, diagnoses or diseases. And we're going to model this as a hypergraph. Now, most of the way in which people predominantly have looked at the connection of new things in the future, predicted new things in the future, is the function of something called triadic closure, where maybe a person connects to other people, or focal closure, where maybe an event or a context, a common shared disease or chemical ends up being the thing that connects those persons over time. And and there's certainly a positive correlation between the number of common neighbors in this space and the probability of things linking in the future. But it turns out that you do much better if you actually model scientists, and not just scientists, but also chemicals and diseases and methods as randomly walking along this entire hypergraph. What this allows you to do is, for example, if this scientist is going to connect with this scientist in a new collaboration at time t+1, then the likelihood of moving through a commonly studied disease, a commonly co-authoring scientist, chemical, or method ends up being, in some sense, inversely proportional to the degree of each of these things, right? So, for example, if this author here, or this institution through which these two scientists are connected, has many, many other people that they collaborate with, or if this method is something that many, many millions of other scientists use, then the likelihood that these two scientists are going to connect through that thing is very low, right? And so allowing us, so essentially modeling random walks along the space allows us to take into account that intuition, basically, that certain elements end up being things that are unlikely to connect people because they connect so many things. And so we model this explicitly and identify essentially random walk similarities between things linking up in the future as a function of these random walk similarities, and identify and are able to predict about 95% of the new connections that are made between any particular thing in this space as a function of these random walks. So we're able to only predict about 50% of all the new ties in the future. So, I mean, every kind of new tie, right? A new tie between a chemical and a chemical, two new diseases, a method and a disease. As a function of these random walks, we're able to capture about 90 to 95% of these new ties in the future. Whereas looking just at these common neighbors, you're only able to capture about 50% of these ties. And the interpretation, so I should just say, kind of, in conclusion to this early part, so the argument is that, papers themselves do a remarkable job of predicting the future papers that are going to emerge in the scientific scene. So it's very difficult to imagine that scientists are using many other things besides papers that are driving the intuitions that drive the next next round of scientific investigations. I'm going to take just a minute to kind of interpret these models. So basically what we're doing in this model is we're using these random walks and the likelihood that they walk through some third thing to predict these outcomes, these linkage outcomes. And one way to interpret this is that essentially, this system of science is a brain, and that random walk similarity scores are acting like a perceptron, basically. So they're basically the likelihood that two chemicals connect through another chemical, or an author, or a shared disease, or a shared chemical end up having weights. So basically, we're kind of using a logistic model, a simple logistic model, to kind of assess the probability that this perceptron fires. That basically people actually, these two things become linked in future papers as a function of 1/1 plus the exponential of these random walk similarities. And what we find is that even though those random walks end up being very predictive, if you look at the basically the weight that science as a whole uses, basically layers on top of those similarities, you find some kind of interesting and unusual patterns, right? So, for example, if two chemicals are going to connect, or two diseases, or two authors, or two methods in some future period, it's much more likely for them to go through some other thing, right? So it's much more likely for two chemicals to go through a disease than to be discovered through a third chemical. Or if a chemical and a disease are going to be connected in some future period, it's much more likely for them to go through something else, right? Through a shared author or through a shared chemicals or a shared method. Or if an author, for example, is going to connect to some new topic, it's much more likely to go through a different type of topic. So if they're going to connect with a chemical or a disease or a method, it's much more likely for them to go through something else. And so this, suggests that scientists aren't exactly like random walkers in this space, that they have this preference for things other than the things that they're studying. And this could be the result of just the structure of the institution of the paper, right? That, the paper kind of needs to have some diseases and some chemicals and some methods. But it's also likely a function of potential bias, as scientists are kind of, once they activate one of these categories, then it may be that they just bias against considering others. We then go on just to try to analyze the degree to which these perceptrons capture the nature of scientific thinking. So the degree to which these weights that I'm talking about end up corresponding with the things that scientists actually do in these papers. And so we analyze the degree to which those third things that we're suggesting are the most likely paths through which science connects new elements are, in fact, those paths end up getting collected in the resulting paper, right? So if I'm positing through this model that that a researcher connects to a new disease through a method that researcher uses previously, that method ends up getting collected in that eventual paper that they show up in together. And we're able to predict about 80% of those collections, which suggests that scientists are in fact kind of walking along this hypergraph of science as they think through the next experiment. Finally, this just shows the likelihood, the global likelihood that they'll kind of connect through one thing or another. And so it turns out that methods and chemicals, which are kind of often in this space like mini methods, end up being really the primary things through which scientists connect to new objects in the scientific space. So anyway, so this is just as a preliminary to argue that science, published science, itself can be seen as a neural net, that scientists appear to think through the literature by wandering locally in this space, which accounts for 95% of new ties between things. The scientists associate between layers and biased against within layer associations. So chemicals connect to diseases, people through chemicals, etc. And that methods are special, most prominent, and the most prominent path by which things become connected and collect along these paths, that scientists are effectively one-trick ponies. So the reason why this is consequential scientifically is sometimes, if scientists are thinking through the scientific literature, then if science is trying to do something besides think through the scientific literature, like solve problems, then there might be a problem, right? So for example, this is a paper that I published last spring, which shows that there's really zero correlation between the burden of disease, so the disability-adjusted life years that disease costs for individuals, and the number of papers that are generated on each of these topics. So basically, science is responding to the prior literature and not to the underlying problems that the literature is supposed to be responding to. Now, one of the reasons that's the case is because, well, some countries are richer than others, and those richer countries tend to produce more science, and the profile of certain disease categories in those richer countries end up being systematically different, not surprisingly, like musculoskeletal disorders and cancers end up being associated with countries that have people that live to old ages, and so those are places that are hyper-sensitive to funding, whereas infectious diseases, respiratory infections, perinatal problems are associated with poor countries and are much underrepresented in funding. If you take clinical trials and publications and burden of disease, you see basically the publications that are correlating with prior publications very highly, but they perform very lowly, have very low correlations or in some cases negative correlation, even within country with prior burden of disease. And so we've created a series of metrics to analyze these. And so you can see with any particular disease, for example, in this case, breast cancer is extremely overrepresented in terms of the number of trials that are kind of performed on this, clinical trials. But as a result, it has this very low kind of opportunity index, which is to say it's much more studied than it is as a problem, right? So if it were, and so you can identify, and almost all of this bias is essentially pushed as a result of the publication system. So NIH granting, for example, in the US, ends up being extremely tightly tethered to publication and publication to NIH funding, which creates these conditions like breast cancer and cervical cancer dysplasia, which are extremely over-studied relative to their burdens, whereas other disorders like extremity atherosclerosis and uteric kidney stone disorders and a variety of other things show much greater opportunity, even clinical opportunity. Although it turns out this imbalance overall has been reducing over the last few years. So the next step in my, so I'm just going to, so basically in this particular research direction, we're trying to add a few things to to study kind of more completely the way in which new papers emerge. And in fact, I'm working with a group of computer scientists at IBM, their computational creativity group, to generate new patent prospects, for example, as also new recipes. To essentially generate full, hyperedges, full new proposed elements and experiments. So we're in the process of doing that. We're also adding features and objects to these random walks and predictions, more than just methods and chemicals and diseases and authors, to really capture the full institutional structure of science. And we're also designing some ex-ante experiments to identify these heuristics that we've identified as biases. Okay, now that I've attempted to establish that science thinks along papers, even when it shouldn't, basically. Now I'm going to kind of use this system to explore the pattern some of the patterns by which scientists choose the next experiment. So how is it that scientists choose the next experiment? Well, there's a whole host of possible factors, right? There's past interests and training and serendipity, the fact that you show up to this particular seminar accidentally. And yet at the same time, I would argue that many of these are structured and modelable. I mean, even though you showed up at that seminar, right, it may be hundreds of % more likely that you showed up at that seminar than somebody else, that somebody else in your field. Of the many things that we could study in this context, I'm going to explore the likelihood of following the crowd versus standing alone, which ends up correlating often with professional productivity versus the possibility and the potential for outsized, surprising, and field redefining outcomes. And the intuition behind this analysis is going to be that effectively novelty assessments, both my novelty assessments and my argument of how it is that individuals assess novelty within the scientific field, is that novelty is proportional to the probability of discovery, right, conditional on prior knowledge and the normal process by which discoveries move from knowledge to the next paper, right? So the idea here is that, if the probability of discovery given prior knowledge in that process is high, 1 minus some extremely small quantity, so it's almost completely probable, then there's very low novelty. And if that probability is very low, then it's high novelty, right? So basically I'm arguing that people are cognitively simulating the process by which something becomes invented. And if it's very unlikely for that invention or discovery to take place, then it's sufficiently surprising that they weight it with a high novelty score. And I'm going to talk at the very end of this talk a little bit how you can use this strategically in the generation of novel products. There are a host of measures, both in biology and in the social sciences, by which people have analyzed these measures of novelty. All of them kind of come down to an implicit simulation of this process. So I'm going to argue for an essential tension in the context of published science, that this getting a publication, getting a job, getting tenure, sticking around is the stable, following the path option versus the Nature paper, the early career award, the Nobel Prize as the kind of the high risk other approach. And I'm going to model this in two ways. First, I'm going to model it by cutting scientific discoveries at their joints. First, by representing them as a graph, and then kind of cutting them at their joints and categorically analyzing different styles of science. So you can imagine that if you introduced an entirely new object into the scientific space, I'm going to call that a jump. You jump into something that wasn't there before. Alternately, you can make a new connection between established elements in the system, or you can repeat an established connection. Moreover, you can connect two things that weren't connected, or repeatedly connect things within subfields, or bridge between subfields. And so again, as with the last paper, I'm taking a series of papers, A, B, and C, and in this case, in this context, I'm analyzing the chemicals, both macro and small molecules. Right, so D, E, E, F, G, G, H, I are all chemicals. Rather than doing a citation analysis, which we also do, I'm looking at basically the connection between these chemical elements based on the number of experiments in which they show up over time. And so using very similar data, about 200,000 nodes and 85 million links. And not surprisingly, this network has a very long-tailed degree distribution, right? So there's a few connections which are discussed tens of thousands of times, and most which are discussed very few times. The same with the number of repetitions, right, of any particular tie between elements. So as I'm cutting this taxonomy at its joints, the one thing I haven't mentioned, so the introduction of a new chemical, the linking of chemicals within an experiment or in some logical pathway. The thing I haven't established is how to identify these subfields. And I'm going to use an approach similar to what I mentioned before, using effectively a random walk approach on this network of connections to identify what I'll call optimal zip codes, right? So the context in which you want to maximize connections within zip code and minimize connections between zip codes provides a two-layer description of these networks which characterize the likelihood that you're going to wander between these nodes rather than wander kind of between subfields of these nodes. And they really, when you do this, you identify core movements in the history of biology, right? So the kind of emergence and separation between the 80s and 90s of DNA and molecular biology from core biological chemicals, and in fact, the eclipsing of DNA and molecular biology over core biochemicals by the mid-2000s. You also see the emergence of things like the second messenger system and the really remarkable stability of things like neurotransmitters and neuroscience, the immune system, hormone chemistry, etc. So these are the fields, basically, on which we calculate these measures. So basically, I'm using these to kind of create a taxonomy of strategies in science. And what we find when we calculate them over this entire period is that they're remarkably stable. From the period that we're able to calculate them, they're remarkably stable. Basically, repeat bridges and consolidations represent about 45% each of the fields of science. New bridges represent about 10%, and new consolidations and jumps represent about 2 and 3%. What's remarkable about the stability is that over the same period, you have this explosion in the number of chemicals that are studied, and an explosion in the number of opportunities that scientists have to bridge between fields. So when we create basically a generative model that matches this model with parameters that correspond to the preference for repeat versus new connections, the only way really to explain this is that scientists have an extreme increased preference for focusing within subfields rather than across subfields over this period. We also find that basically the surprisal, or the rareness and difficulty of the strategy ends up being associated with higher citations, both on average, both with mean citations, and it accounts for a greater amount of the variance in citations over time, which is to say that jumping to new chemicals also has both a higher and a lower opportunity, but a higher expected value in terms of citations. Now, one thing that's not captured in this space is the number of jumps, for example, that were tried and failed and never made it into publications, right? This is a sampled set. And so we use that to kind of ask the question, well, it turns out that observed investment in riskier strategies can't be rational if scientists are just trying to maximize citations, because if they were, new strategies would have only 24% and jumps only 35% more risky than just repeating old strategies, right? Which is to say that there's some combinations between distant elements that just should never be tried, and that fail, like this connection between sandwich and canning technology, which failed before it hit the market. Sad for us. So what does motivate risk-taking, right? If citations, right, if scientists, if they were to maximize citations, would actually be even more risk-averse than they are. What about exceptional achievement or highly cited papers and prizes? So we matched all these strategies to basically every global prize in biology, chemistry, and medicine over the last half of the 20th century. And we find that, so this is basically the average across all papers. And these are the top 1% of citations in terms of their enrichment and jump. So they're more than 200% more likely to introduce a new chemical if they are in this top 1% of citations. If they're among Nobel Prizes or the pre-Nobels, prizes which predict Nobels, they're somewhat more likely to have introduced new chemicals. They're also much more likely to have new consolidations, which is interesting. These are new connections within existing fields, right? So there's two things going on here. One, if you have two things that are connected in existing fields, you have an audience, which is typically an awarding audience that can recognize and appreciate the thing and therefore grant it an award. It's also more likely that those connections have been tried before, right? And so it provides a quality metric effectively, because if there are more people who have also been aware of previous trials, then it's likely that it was a harder connection to establish. But so this suggests that that scientists, even though they're not sufficiently motivated by citations, essentially to kind of move into these nether reaches of extremely risky strategies, that it's likely that these elite prizes and extremely high citations, so kind of outsized fame, end up likely kind of nudging them in the direction of the strategies that they analyze. So next, I'm gonna examine. So we've kind of talked about, published science as basically tracing the way in which people think and the liabilities, some of the liabilities associated with that. We talked about one aspect of the scientific system, which is the mix of kind of risk and reward. And here I'm going to kind of explore the idea of the efficiency of this search process for uncovering what it is that science has uncovered. So this demands a more highly resolved view of the network rather than just kind of creating a taxonomy where we cut it at its joints. We're going to have to create a generative model which kind of tries to capture and measure the strategies by which science as a whole and individual scientists uncover this network over time. So, and we're going to consider in this model that scientists themselves consider this actual network of ties in their decisions of what to experimentally combine next. And in particular, they're going to consider the centrality of certain elements, certain chemicals in this network, right? So these really correspond to highly important, highly repeated chemicals. And we're also going to argue that they consider the distance between them in this space. So they consider whether or not they're in the same subfield, and really, in this case, how directly distant they are from one another. And you can trace out basically any combination of chemicals across papers, right? So this 2000 piece, which connects estradiol and cholesterol, which is basically the paper that nullified the idea that the hormone therapy was effective in battling breast cancer in women. So both of these things end up being, highly central in this space. And zinc and RNA, this is the discovery of zinc fingers in 1985. Here's one where you have a very different pattern, right? So you get DNA, and obviously highly central, that connects with bromodeoxyuridine, right, which is the therapeutic, but then was used in its traces in the brain to identify the kind of mature growth of new neurons in the brain. So basically, we're arguing that scientists are aware of these distances and centralities as and in fact, they characterize strategies that scientists themselves think about over time. In fact, you can also see that they're basically, as you look at these chemicals, these are chemicals that are connected across the years until in this one paper in 1996 that rapamycin and genomycin D end up getting connected in this particular paper. But people are really thinking and connecting geographically, right? So people are aware of each other's work in Japan and in Germany, and that essentially these are, they're geographic networks that essentially are being traced by these chemical networks. So if we just look at the raw data, this is basically the relationship between any new chemicals that they combine. They both tend to be highly central on average. So this is the kind of a density plot. Although if we look at the citations that accrue to those citations, this is the mode of that distribution. So these are both highly central chemicals that are being connected. They're much less likely on average to accrue high citations than those which are essentially non-standard tree distributions. And if we were to map those prizes that I mentioned on before, all of the chemicals associated with, or chemical connections associated with prizes, they have a much higher entropy degree distribution. So it's much more likely for those prize-winning papers to connect things that are much lower frequency distribution. If we look at the distribution of distances over time, prizes also end up being more likely to be completely disconnected in their kind of network components over time. And we see basically that distance, the distances stay relatively stable over this period, although these finite walks, so this is the number of infinite distances or jumps, in terms of the finite walk between chemicals in the space, they actually become smaller and smaller over this period. So this is the model we're going to squeeze all the variation of this network into. So basically we're going to say that a scientist's probability of connecting two chemicals is proportional to basically the centrality of each of the two chemicals that they're analyzing, and basically a beta distribution of the distance between them, right? And if they're of infinite distance, then we have an exponential parameter corresponding to that. When we look at the results, when we estimate this model over time, we find that science becomes more and more conservative from 1975 to the present, right? So basically, they pick more and more central chemicals to analyze over this period, which have a smaller and smaller distance between them over this space, with a greater anti-preference for jumping between network components. So this is the direction in which this network actually goes, and we then take a large distance and degree distribution subgraph of this network. And then we basically re-estimate not only this model, but to get at the idea of efficiency, we walk through the parameter space, right? The parameter space of this model. So this is the alpha mu and alpha iota are the preferences for the centralities of the two chemicals, the most and the least central chemicals. Negative beta, negative gamma, and delta correspond to these distance preference parameters. So what we're going to do is we're going to take this large random subgraph and we're going to sweep through the parameter space and identify more efficient strategies of discovering what was discovered, right? So this is one of the random samples that we took out of this space, and then this corresponds basically in time to the strategy that is performed effectively by the scientific system, so that you see across both Medline publications. We also estimate this for U.S. patents, which provides an almost identical trace. So what you can see, again, is that basically a few chemicals become really central hubs. We estimate this over about 500 times in each of these instances, so that everything's connected here, but certain things get hyper-connected, and many things are underexplored until very late in the discovery of this network space. We find that the existing strategy, what I'll call the Medline strategy, the strategy that you induce science is following based on the stream of publication, is about the most efficient for discovering 10% of the network that becomes discovered in the end. But it's unbelievably inefficient at discovering the whole 100% of that network. You do much better with, 100% strategy, 50%, a 20% optimal strategy. I hate numbers graphs, but I actually think this helps tell the story. What's going on here, if you estimate the optimal search strategy for each of these approaches, is that if you want to uncover about 10% of the network, you pick a hugely central component and you just look at its marginal connections, basically. But they're diminishing marginal returns to theory, right? So once you understand that structure, if you keep exploiting it, if you just keep picking out what you think of as the central elements over time, that's going to do a very bad job at uncovering 100% of the network. So optimally, basically, you have a smaller and smaller preference for the centrality of these chemicals over time, and a greater and greater preference for risky connections between more distant elements. It's kind of like if you were to think, when's the optimal time for, somebody to kind of drive fast race cars, jump out of airplanes, and it's not between 18 and 25. Right? It would be ideal if they started that at 80, right? When the life years that they lose as a function of taking those risks are largely gone. So what's interesting historically here is that this is precisely the opposite path as what historically occurs, right? So in kind of uncovering this search space optimally, you go from most conservative, from exploiting all the structure you're aware of, to the least conservative, to kind of jumping to disconnecting components and really capturing the fractal elements of the network as we observe it. Historically, precisely the opposite happens. Scientists become more and more conservative, jumping shorter and shorter distances. This is, for example, the optimal 100% network. It looks like a random path, but it does much, much better than random in practice. So basically, if this is the Medline US patent space, this 50% optimal search ends up being much less likely to kind of follow this swarming herd mentality that science demonstrates. We also tried to kind of imagine another world, which corresponds really in many ways to kind of company research, where there's a command and control structure and no experiments are repeated. right? So one thing that we have as a problem in some ways in the scientific space is that for the most part, negative knowledge is not published, right? So actually in these chemical connections, we've parsed them out much more precisely than just looking at these chemical associations in the context of molecular biology. And about 5% of the connections which are published are negative associations, right? So protein A does not combine with protein B under some condition, right? So there aren't publication outlets for negative knowledge. But within companies, within institutions, that negative knowledge tends to be remembered, right? And so we just imagined and analyzed a world on the space of actual publications and findings of the benefits you would get if you coordinated, which is to say, if you performed every experiment only once, given the parameters that we've uncovered, and you end up doing much, much better in the case of the optimal coordination, at least an order of magnitude better than the kind of the Medline uncoordinated strategy, which is the strategy that we observed in the data. Obviously much better than random search, and that even if you just took the existing Medline strategy, that you would do about half an order of magnitude better, of course, increasing at the end, right? As you get closer to the end of discovering the whole network, in the context of Medline, you have this huge iceberg of unpublished findings, which stay, hidden and discovered over and over and over again, essentially stopping the scientific system from kind of moving on to resolution. So where do we go next with this? Well, we're doing some experiments. We're doing ex-ante prediction of risk and accuracy in pools of genomic experiments. We're adding richer models of human behavior, and we're extracting other units and links of combination of chemistry, biology, math, and other spaces. Now, one thing that I haven't mentioned so far is that why am I studying this in the context of chemistry and biomedical chemistry? And it's because the nouns and verbs are so well behaved, right? So, chemicals, reactions, both they're extractable, but also the kind of the precision of the use of those units ends up being very stable over time. So I did another analysis just to kind of consider the relative precision, which is to say how much harder would it be in another context. So I take this information theoretic notion of entropy, and which you can calculate in the context of flipping a fair coin. It's maximal, right? So basically, it's the idea that you're completely uncertain. If you're completely uncertain, then you have maximal entropy. In this case, one unit or one bit of entropy. Whereas if it's an unfair point, if it only turns up heads, then you're absolutely sure of that random variable's outcome. So one can think of the same thing in the context of the synonym substitution probabilities of a given word or chemical in a space. And what you find across fields is that it's true. Biology, medicine, and chemistry basically have the lowest entropy rates for these terms, right? So they are, in fact, the best behaved. And then it becomes much trickier as you move out to the physical sciences and mathematics. This is, we haven't calculated the equations here, but we're in the process of doing it. Social sciences, law, business, management, and the humanities are way down here. And in fact, if you look at novels, so these are British novels in the 19th and 20th centuries versus biology and medicine versus the news. You find that basically novels have the highest entropy, the most metaphoric and diverse usage of elements. So stretch, for example, being used as open, spread, draw, completely words that don't themselves share each other as synonyms, whereas increase, develop, enlarge, and prolong are those in medicine and biology, which have a much lower entropy. It's much more likely for any one of them to be the substitute. that being said, we also have kind of used this approach to kind of identify what are the things that are missing in these synonym dictionaries. And a piece that we published just a couple of weeks ago in PLOS Computational Biology, we identify essentially the size of these missing synonym dictionaries, and we kind of recover a number of these missing kind of commonsensical synonyms through a series of crowdsourcing experiments, which end up being quite effective. We're also in the process of extracting other entities, like these equations, for example. So we're taking from the LaTeX these equations themselves, re-rendering them as graphs and identifying them effectively as conceptual units. And we're able to analyze these then in the same way in the context of the mathematical and physical sciences and get a much richer analysis of the way in which these fields combine their elements. We're also extracting figures from these fields to identify common underlying pipelines that characterize the way in which science goes forward. So all these things combine to say, so how does science think? Well, I'm arguing that it thinks through different types of things, that methods furnish a special connector by which new elements are combined, that there's stable institutionally transmitted research habits of combining things within subfields that have been repeated over time, that tends not to be sensitive to changes in the space of possibilities. In fact, it sets up barriers to exploring this possibility space. But there really is a kind of Kuhnian essential tension that's driven by imitation of awarded work, and that's efficient for discovery of about 10% of what we know, and then it could be much more efficient if failures were published in this space. And I think this also, this holds out some possibilities for policy and for encouraging high-risk work, right? So there are contexts like Bell Labs and Howard Hughes Medical Institute, where you're decoupling productivity from job security, where you're funding people and not projects. And in fact, this, based on our and several other recommendations, has really been, there's a whole new series of grants that came out just last year in NIH to do exactly this, to fund people rather than projects. Lowering barriers by funding risky projects more aggressively themselves. And there are also efforts it also suggests ways to encourage the publication of failures. Things like clinicaltrials.gov. So this is a U.S. institution that forces essentially anything that results from NIH funding needs to be published. It turns out only about 15% of the things that have been completed in these trials end up actually getting entered to this database. And so so there are legal efforts afoot that I'm supporting that encourage the federal government to essentially use the policies. There's a $10,000 per day fine that's never been levied for these lack of compliances. And there's also incentives for the production of negative knowledge explicitly. Now, finally, I'm just going to mention for a few minutes how this knowledge about knowledge can enable machine science, right? You can kind of use these identified biases and heuristics as instruments, basically, by which you can go forward and do better at kind of predicting clouds of hypotheses. And in fact, insofar as you are able to do better, that provides a deeper validation that you actually understood these heuristics as biasing, right? If you don't, if you can't do better taking them into account, then I'm dubious that we actually knew something about this process in the first place. I'll just sketch out one analysis that we're doing currently in the next 3 or 4 minutes, and then I look forward to questions. So this is, we're engaged, myself and a handful of collaborators are basically taking a series of cancers, and we're looking at them from publication all the way through a series of robotic experiments to kind of optimize the clinical dosage of basically cocktails of therapies. And so in this process, initially we kind of extract these gene-gene combinations to identify underlying pathways, and so these are pathways associated with A cancer and a couple of neurological disorders. And then we're taking that understanding of the scientific system and we're weighting these claims, right? So we can show, we've shown in other work basically that scientists attach and follow these central things, right? As we showed earlier, they're much more likely to connect things that are formally connected in papers. But it turns out that those repetitions don't always correspond either to independence, accuracy, or take into account the selection of those findings within journals, right? So basically, we model each of these elements, right? So we model the independence of the evidence by taking into account the number of independent methods which were associated with each particular finding, the number of independent authors and clusters of authors that were associated with each finding, the accuracy of investigators that we model as a function of essentially their agreement with the other class of investigators, and then the selection of findings by modeling both the novelty. So if one thinks about the production function, the likelihood that a paper gets in a journal of a given status as being a linear combination of the surprise associated with that discovery and the support associated with that discovery, right? So the idea is if something makes in nature, it's either got to be really surprising or it's got to have, enormous support or some balance of those two. And we can demonstrate that over this context. But then if you do that, you can have different measures of the support of classes of evidence. So for example, the intuition here of independence is that a finding wouldn't be accorded at, or shouldn't be accorded as much clinical strength if it's performed by, one PI and 3 of her students using 1.5 independent protocols, as if it's been analyzed by 10 socially independent PIs using 5 different separate protocols, right? So even if those things show up in the literature the same number of times in the same number of outlets, you would argue that one represents fewer draws from the urn of truth than the other, right? So taking these things into account, then you reweight this network of science to kind of correspond with how much empirical evidence actually is associated with it. We've actually done some studies which show that the support that we receive for subsequent reanalyses of these findings corresponds to our reweighting of this graph, right? So even if things show up many, many times, but they're not independent, and they're by investigators that tend to diverge from other investigators on average, and that they have low support based on the inverse probability of the surprise, then they end up being findings that are less likely to show empirical support in the lab when we rerun them. And then we take other data and we filter all the range of possible mechanisms by this exogenous data. So, for example, in this case, we gathered about 110 million people's clinical records. So this is about a third of the US population. And we have, for those people, so these are from insurance records, diagnostic codes over time, procedures that were performed on them, and drug codes that were administered to them, right? And so you can kind of lay all of these on top of a series of molecular mechanisms which are reduced to the literature and reweighted according to our understanding of biases involved in the production of science, to identify the maximally informative experiments that can be performed to identify plausible drug combination outcomes. I'm just going to show a couple of other things that you can do with these large clinical data that end up constraining the space of clinical experiments. So, for example, this is, we analyzed the Granger causality associated with basically every clinical category in this database over the course of about 15 years, and identify things like the difference between viral infection, right, across this space, which typically comes from the coastlines inward, and fungal infection, right, which is very different itself from something like Staphylococcus aureus, right, a skin-to-skin infection. And so you can use these kinds of data to constrain the space of analyses that are performed, and we're now in the process of computing all of those treatment approaches to identify the likelihood, for example, that treatment X on diagnosis Y ends up leading to outcome Z. And so then we layer those, basically all of those natural experiments, all those natural clinical experiments on top of the laboratory data to constrain the space of experiments that we study. Finally, then we estimate the surprisal, right? Or I think more generally, the Bayesian surprise associated with we're estimating what experiments are going to be maximally informative given basically the former literature and this constraint based on enormous numbers of essentially clinical experiments from the medical establishment. And so the intuition behind Bayesian surprise is that you're basically taking, well, Bayes' rule, what's the probability of a model given data, so that is really equal to the probability of data given a model times the probability of the model over the probability of data. And so basically the surprise associated with any particular, the clinical surprise associated with any particular experiment ends up being, or we calculate it as the Kullback-Leibler divergence between the probability of the model when it's updated by one datum, right, by that experimental datum that we're analyzing, and the distance between that prior model before it was updated, right? And so it's been shown in other work that this Bayesian surprisal corresponds with what individuals themselves experience and attend to as surprise, right? So these are a series of priors, and basically, based on your experience, controlling for that prior experience, then you can calculate the surprise differently based on the perspective that individuals have. And in a series of analyses of basically eye tracking and movements, people attend to surprise in ways that's ideally captured by this analysis. And people experience surprise in novel recipes as a function of essentially the Bayesian surprise of those flavor combinations. That was just optimizing olfactory surprise, conditional on pleasantness. So the idea is, we're using right now these surprises to kind of identify optimal experiments to run in a very expensive case where we're actually doing human cell tissue culture analysis to identify optimal drug cocktails. So the idea here is, can we create better therapeutic cocktails by understanding the system of science better? So I'm going to close with that. Thank you for your time.
So we have about 15 minutes for questions. I think we need to wrap up at 4:20 because some of us have to be in another meeting at 4:30. So I think, well, you can probably handle your...
Sure, yeah, this is an intimate group. Please.
Which of your findings are the maximum surprise?
Which have the maximum Bayesian surprise. I need to factor in your personal background to really. So the question comes back to you. I think, in some ways, I think this is not that surprising. I mean, the idea that the system of science, the complex system of science, people attending to other scientists, engaging in these kind of local crowding effects, attending to kind of things that end up showing up higher in the literature, and accounting for those things can allow you to do better, is not that surprising. And in some sense, but for me to prove that to myself and to others, basically, I need to reconcoct those experiments, taking this into account, and see how much of a delta it gives us, right? So, so in some sense, my argument is that that really this is a completely, banal project. That basically, if, and in fact, I run a network called this Meta-Knowledge Research Network, and I fund projects in this network, which is composed of about a third computational scientists, and about 2/3 domain scientists in everything from the genomic sciences to cosmology to ecology, for scientists to take into account essentially the complex history of questions to identify better questions for their disciplines. And so I hope it's true at the end of many of these studies. I just gave out about $1.5 million about a week and a half ago to a bunch of different small groups in seed projects. My hope is that it will be even more unsurprising to find that they can find better questions or concoct better questions by taking into account essentially the path dependence, the swarming, the kind of the dynamics of the system that we're uncovering. So,
amazing things and also surprising things is that it's kind of the knowledge based on knowledge type of knowledge. Does that give some recommendations? It could be completely separate from the substantial knowledge of the discipline itself. So for me, this is really surprising, right? As I'm not sure whether this is because you use science embodied in publications, and that is the difference between the practicing science every day. So there is a lot of like the intuitions and uncertainties and different judgment in the process is basically it's not the final publicized evidence.
Correct. Yeah, and in fact, I would say this is a kind of a core element of this project is that, so while the publications trace a whole series of scientific actions across the space, it doesn't itself explicitly embody the theory of search by which these fields analyze their questions. And it turns out, and maybe this is an answer to surprise, is that many of these search processes are similar across fields. And I haven't demonstrated that here. I've just shown for chemistry and for biomedical sciences, but I can show in a number of other fields as well with some of the more recent, some of the more recent similarities between equations, that similar games are being played in other parts of science. So in fact, that's one of the bits of surprise that I'd like to really nail down going forward, is exactly how widespread certain of these tropes or these movements are, these kind of hidden theories which capture the practice of science, that taking them into account, we can do better, right? Because essentially they steer science towards certain questions and away from others which were equally or more promising. So, I would say tenure is one of the problems. Yeah, I think, well, one thing that I think is really striking is, so why is it that scientific investigations look like this? Well, they look like this because individuals are optimizing their own evaluation, at very local timescales. And so while it is true that the optimal experience for science at any one point has a 1 in 10,000 likelihood of coming to pass, and individual scientists may not be able to amortize that risk. One way to change that objective is to evaluate groups, So I think this is one place in which our scientific evaluation system is just supremely poor, is it really evaluating intersubjective group work. And places like, Bell Labs in its heyday really did beautifully, right? So they allowed failure, and in fact they expected and rewarded failure indirectly in a number of ways. So I think one way to consider this is actually placing some value in the context of a tenure system on group outcomes, right? The likelihood, the distribution of outcomes from a rewarded unit ends up. So I think that's exactly the reason you see this pattern.
One question I was just thinking about is where the bottleneck is. Right? You can imagine several. One is that people just from the beginning are picking safe topics, right? That they're going to publish the 100th paper on this particular topic, right? And then there's another possibility is that people are having innovative ideas, but then when they write the paper, it gets rejected and it's never published, and then they don't get in here, etc. So I'm saying in terms of figuring out where the bottleneck is, are there opportunities to do? I imagine sort of studies focused on specific pieces, and sort of, if you could get like the complete corpus of an change submissions, right, in the abstracts, and then you could look at, do novelty measures for the abstracts of the submissions, and then look at whether
The funded and the unfunded ones.
Yeah, and predicts even the scoring. Is that some, or even journal submissions?
Absolutely. So I just got
the full submission and review process data for PLOS One over the course of about 3 years. So, who's published in PLOS One? Anyone? Okay. So basically, yeah. So I've got all the submissions, all the changes, all the reviews, all the identities, which again, it's all in some secret repository that I throw my algorithms over and link and then bring them out. But we're able to identify exactly this notion of, so yes, things that are more likely to be surprising or more likely to fail across the review process. Things that, but we are also able to capture this Bayesian surprise element, which is to say that things which are distant from an individual investigator end up having a higher variance of review, either being completely wacky or completely mind-altering, and highly successful, right? Whereas things that are much closer to the reviewer are much more likely to have a stable outcome. So we're able to see some of these patterns, and I think one of the ways in which I'm trying to get at this filtering problem, right, is by doing these ex-ante experiments, right, where I'm able to generate kind of a series of questions which has been asked by the scientific system, for example, in the context of yeast biology, and then I'm able to generate a series of alternative possible questions that would likely have been would maximize the information value of each experiment and see if those do better, basically, in the whole essential interactome of yeast. So this kind of provides a way of kind of getting all the negative results on the table in the same class of experiments as the positive results. And it looks like we are able to do better in the experiments that we've done. So,
I kept thinking about your 2010 paper on how the industry needs scientists to know less about more. And what struck me is that that the industry changes how scientists make scientists less conservative, opposed to what you say today, and change the scientific collaboration networks. But what I don't, what still seems to be a puzzle to me is that when this more community of scientists collaborating with industries and doing more speculative, more risky experiments emerge, are they sanctioned by the overall scientific community, and does the scientific institution and community have the power to sanction them? If so, why the institution, why the emerging institution of doing scientific work, doing more risky, speculative scientific work could emerge and get to be so prevalent today in the United States and in the world? I know a lot of institutional sociologists are doing research on this project, but do your network analysis of over time trend could provide some answers to this question?
So that's a big question and relates,
predominantly to previously published work. Thanks for reading. But I think this work suggests another reason why it is that companies are likely to fund this kind of work, right? So one is they don't have the commitment to basically priors being connected to posteriors, right? So the likelihood in the context of a grant funding submission relies on, well, historically at NIH and these other funding agencies, and I should say that's very different from funding agencies like at least in the US, the defense-related funding agencies, which are explicitly optimizing surprise. But in the scientific funding agencies in the US, so Department of Energy, NIH, NSF, these agencies have historically been really quite conservative, and increasingly so as the years have gone on. And industry has a couple of, one, they in some cases have no special like they have an orthogonal interest in a particular interaction. So for example, if a certain relationship ends up showing up true and is clinically relevant in a biomedical company or kind of industrially relevant in like a plant biotech company, then whether or not it was expected to be true, right, scientifically is just gravy. So they weren't betting on a publication, they were betting on a product. So I think, I think another thing that we see the industry does in the context of the work that I presented here today is they do something that academic institutions tend not to, which is value the total productivity, basically, of groups and teams. And so that's another way in which individual scientists can engage in highly risky products, where the company can act basically like a venture capitalist, right, in expecting only 1 in 10 to kind of turn out. So basically the collective funding and evaluation structure of industry allows them to engage in risk in a different way. And what you see, even in scientific institutions, that larger institutes end up optimizing surprisal in a very different way than individual scientists, right? So if you look at these large genomic centers, they are explicitly trying to kind of push this right-hand side of kind of risky experiments to maximize the surprise value of their discoveries. Although it turns out even in some of these institutions, it's very difficult for them to actually get their postdocs to perform those experiments. Right? So the design a slate of experiments and you look at them at the end and it's like, wow, that's a much more conservative set of comparisons than I was expecting. And you find that they got retranslated by postdoc scientists along the way who were minimizing risk, right, and maximizing the likelihood of a published output. So it may be time. Thank you, everyone, for your time. This has been great.